division of cytoplasm is called cytokinesis, etc. Okay. Here, karyokinesis involves 4 major stages. First one is prophase, metaphase 1, anaphase and telophase. Now, what is prophase? Let us see. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of the interphase. Okay. In the S phase, what happens? And in the G2 phase, what happens? The new DNA molecule formed are not distinct interminor. Prophase is marked by the initiation of condensation of chromosomal material. Initiation and condensation of chromosomal material. The chromosomal material becomes here, okay, untangled during the process of chromatin, condensation. In this case, the centrosome, if you carefully observe that centrosome which had undergone duplication during S phase of interphase, now begins to move towards opposite poles of the cell, anterior and posterior side. The completion of prophase can thus be marked by some of the following characters. Okay, here chromosomes condensation to form the mitotic chromosomes. Chromosomes are seen to be composed 2 chromatids attached together at the center of the chromosome called a centromere, and these branches are called chromatids. What are they called? Chromatids. Centrosome, if you carefully observe the centrosome which had undergone duplication during interphase begins to move towards the opposite poles of the cell. Each centrosome radiates out microtubules called asters. Okay, radiates out microtubules called asters. Okay, the 2 asters together, spindle fibers form mitotic apparatus. In the diagram, I will show you how this mitotic. You can see here mitotic apparatus that are there in it. You can see the mitotic apparatus. Okay. Next, after this prophase, what is their metaphase? Complete disintegration of the nuclear envelope. Nuclear envelope is disintegrated. And second phase, it is the second phase of the mitosis. Hence, the chromosomes are generally spreads like through the cytoplasm of the cell. By this stage, condensation of chromosome is completed. Then they can be observed clearly under the microscope. Here, this is the stage at which morphology of chromosome is mostly easily studied. At this stage, if you take the metaphase, chromosome is made up of 2 sister chromatids. What are they? 2 sister chromatids, which are held together by centromere. Small disc-shaped structures at the surface of the centromeres. There is a disc-shaped structure at the center of the centromeres. They are called kinetochores are also present. Now here, these structures serves as the site for attachment of spindle fibers. What are they? To the chromosomes. Okay. Now, hence the metaphase is characterized by the chromosomes coming to the equator region, and one chromatid, the arm of a chromosome, one chromatid of each chromosome connected by its kinetochore to spindle fibers, okay, from one pole. from one pole and its sister chromatid is connected to kinetochore to spindle fibers from the opposite poles. The plane of arrangement of chromosomes at metaphase is referred as a metaphase plate. Here, the key feature of metaphases are spindle fibers, whatever there, attached to the kinetochores. Spindle fibers are attached to the kinetochores of chromosomes, and chromosomes are moved to spindle equator, get aligned at the metaphase plate and through the spindle fibers at the both ends. What is anaphase? At this onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously into two daughter chromatids now referred to as daughter chromosomes of future nuclei and begin their migration towards the opposite poles. At each chromosome moves away from the equatorial plate, and centromere of each chromosome remains directed towards the pole of, and hence its leading stage, with the arms of chromosomes trailing behind. Now what happens during telophase? At the beginning of the final stage of karyokinesis, if you take the telophase, in this telophase, chromosomes that have reached their respective poles, okay, now chromosomes reach to their respective poles, imagine like this, chromosomes move to their respective poles. Now what happens when the chromosomes move to their respective poles? Imagine this is anterior pole, this is posterior pole. Finally, the individual chromosomes can no longer seen as a set of chromatin material. Okay. Finally, this is the stage which shows chromosomes clusters, okay, at opposite spindle poles, and their identity is lost into discrete elements. Nuclear envelope develops. So here also around these chromosomes, nuclear envelope is developed, okay, forming two daughter nuclei, okay, one nucleolus, Golgi complex and endoplasmic reticulum. Now these are the stages, okay, you can see early prophase, late prophase, okay, transition to metaphase and metaphase stages.